Hello class. So today's lecture is about the cell. So from sugar to gene. So the gene is the section of the DNA molecule. So here you see the chromosome, and chromosome has this sequence of building blocks. Chromosome is made of a bunch of genes, and if we zoom in there, we'll see what the DNA is made of. So the most important thing of a chromosome is this gene. The gene is your hereditary information. The gene is a particular region on the chromosome that carry the message for development of your particular characteristics. And that characteristics is your trait, your phenotype. So the trait or the phenotype, that's how you look on outside or inside. So it could be the eye color, it could be straight or curly hair, it could be your blood type. It's basically your makeup. So also these genes or this genetic information directing a protein manufacture in a cell. And as you know, within the cell we have 23 chromosomes coming from father and 23 chromosomes coming from mother. And each chromosome is divided into a gene, right? So gene, and that particular gene will code for a particular trait. So this is the big picture. Now we're gonna discuss little details. Genetic code is the set of rules that translate information encoded with the genetic material. So the genetic code explains how only 4 nucleotides A, T, G, and C with only 20 available amino acids can synthesize thousands of proteins. Think of this as the letters in the alphabet. So here's this visual example. Like you have different amino acids and they have different shape, right? Each shape relates to a different function. So you put them in a different sequence and they will take a different shape and they will have a different function. Because let's say one amino acid could be positive, another amino acid could be negative, another amino acid could be hydrophobic, another amino acid could be hydrophilic, and they will curve differently. Pluses will repel each other, minus plus will attract each other. So the final product, the protein, might look like this. And with the same available amino acids in a different sequence, another will look like that, and they will have a different function. So with the varieties of those 20 different types of amino acids, we're able to make or synthesize thousands of proteins that make up us. These letters, these nucleotides, we can also refer to them as codons, and the codons will represent 3 of these nucleotides sitting together. C, right? Let's say T, G, and C, and that's a codon. So the multiple codons is a genetic information. So let's look at the chronological order from smallest to largest. So the DNA, the entire DNA, or we call it nucleic acid, DNA or RNA, the building blocks or monomers are adenine, thymine, cytosine, and guanine. And we put them in pairs. A we matched with T in DNA, and G we match with C, also in DNA. If it's RNA, then instead of T we'll have a U, but G and C stays the same. So if you're looking at the DNA, unraveled DNA, and you see these nucleotides, and if there's a U, then you know that it's RNA. So when you take them together and you arrange them in the proper sequence, then we'll have a nucleotide. So the nucleotide will have a nitrogenous base. Let's say one will look like this. Let's say this is T. Then it's connected to a sugar molecule, and it's also connected to a phosphate group. So this is your nucleotide, and the nucleotide in a different sequence, that's a genetic information, that's a gene. So bunch of these nucleotides make up a nucleic acid. So the DNA has a double helix, double strand, and along the strands, that's where you will have this nucleotide. T will match with A, C or G would match with C, and so on. So the DNA is a double-stranded molecule composed of many nucleotides in different sequence. So that's where we store the genetic information in the sequence of its base pairs. So, and finally the gene, the collection of those base pairs or the nucleotides. So that segment of DNA contains the instruction for building a specific protein. It could be functional protein, or it could be structural protein, like structural protein that is part of the cell membrane, and functional protein, it could be a hormone or an enzyme that will perform a particular action. This is the chronological order, but now let's look at the smallest of those monomers, at the nitrogenous bases. So this nitrogenous bases, adenine and thymine. Adenine always pairs with thymine and has double bond. Guanine pairs with cytosine and has a triple bond. You see, like here, G, and C has a triple bond. And adenine and guanine, we call them purines, and the thymine and cytosine, we call them pyrimidines or pyrimidines. It depends on how you pronounce. So, and it is always in the DNA, number of guanine units must equal to the number of cytosine units, right? Because one complements another, and the same thing with adenine and thymine. So here's a nitrogenous base. Let's say this is A or G, and here's a sugar, ribose. It's just the name of the sugar. And here's a phosphate group. So when you look at the ribose, you can tell if it's a DNA or RNA. So you see this ribose here has this hydroxyl group, and the deoxyribose is missing oxygen. So this is DNA, and this is an RNA. Deoxyribose, deoxy meaning oxygen is gone, right? So if OH here, then you know it's a ribose. If it's deoxyribose, like this, then it is a DNA molecule. So this is a nitrogenous basis. So, and when you pair these nitrogenous bases, that will lead to, sequentially, that will lead to a formation of a gene. So now let's look at the bigger picture, since you're already familiar with those building blocks. So if you see there is a double helix, you know it's DNA. So A with T, G with C, and so on. So the collection of these nucleotides, that's what makes a gene. So here's another nucleic acid, RNA. So the DNA is a double helix, and the RNA is the single strand. And you see, instead of T here, we have U, uracil. And the RNA is mostly active outside of the nucleus. And there are three major varieties of the RNA. One is the messenger RNA, another is the transfer RNA, and another is the ribosomal RNA. And we're gonna look at these RNAs separately. So now that you're familiar with DNA and RNA, let's look at something that is called DNA replication. Why do we need to replicate? So replication, it is copy, right? Copy, identical. Identical copy. Nothing will change. Identical copy of your DNA. Preparation for cell division. When the cell is about to die, let's say, and we need to make a new cell, or you have an injury on your skin, right? And you need to make a new skin cells or any other cells in the body. So we need to make a new copy of DNA because you want to copy all those 46 chromosomes into a new cell, and you want to have identical information, because if those cells would not be identical, the new synthesized cells, then you would look different. So we take this original DNA, right? It's just a chunk, obviously, and here's A paired with T, C with G. This is just the DNA, we're not talking about the RNA. So we split them apart. So it's basically what happens here. Enzyme helicase, not that important now, breaks down these hydrogen bonds, and we separate, and now we have two original strands of the DNA that could be served as a code for a copy of DNA. So here we have an original DNA, obviously it's a chunk, So two sides could be a template for synthesizing a new strand of the DNA. So this side, which is original, and the other side, which is also original, dark blue, it's a old strand of DNA, and they will serve as a template, right? So here we're gonna have the new one, right? So A pairs with T, C with G, T with A with T, and G with C. And another old, so this one is new, right? A, C, T, A, and G. So, and then you have an old strand, and then you have an the new one, right? And the same thing on the other side. So it's basically you go from the original, right? You're making a new copy. You're making a new copy. And then you end up eventually with two new daughter cells, right? You start with mother, and then you have daughter cells. And that's what is a DNA replication that we needed for cell division. We'll discuss cell division later, but that's basically what happens during DNA replication. We just take one and we make a new one. So there are two terms. One is the chromatin, another is the chromosome. So what is the difference? It's the same thing. It's just what we're accustomed to, right? So we see this under the microscope, right, as visible, but we do not see DNA in this form. We see the chromosomes when they're ready for replication, when they're ready at the stage when they need to be divided, right? So when we'll pull one side of this chromosome called sister chromatid and another side of sister chromatid to their respective poles, but we will discuss this also later. So the chromatin is just the same DNA, only it's unraveled. So in the nucleus, right, they look like that, all 46 chromosomes, 23 from mother, 23 from father. So, but when the cell decides that now it's time for a cell division, right, so DNA needs to replicate, and we're making a copy, and then under the microscope you're able to see them in this formation, right? Because eventually one side will go to one daughter cell and another side will go to another daughter cell. That's how we create a identical two daughter cells. But then again, that would be later. So I just wanted to touch on chromatin and chromosomes. So let's say uncoiled, and then it is a condensed, right? So we call them sister chromatids. It's identical replica of the original one. So now let's talk about protein synthesis. So we just discussed DNA replication when you make an identical copy in preparation for cell division. But let's talk about protein synthesis. So we also call this a essential dogma of life that includes transcription, translation, that will lead to protein synthesis. Why do we need this protein synthesis? Well, you have a damaged cell, we need to make a new cell, or we need to make a structural protein that will be used for supporting a cell properties, or we need to make a functional protein like hormone or an enzyme. So this DNA, right, this double helix, carries the code for protein synthesis. Yes, it's your genetic information, but also this DNA carries a code, just a bunch of nucleotides, which are genes, that will code for a particular protein. And here you see the entire process. So this process starts with the nucleus. Within the nucleus we use the DNA, we use this genetic code, those nucleotides, those base pairs. We copy the DNA, not similar to a DNA replication when you copy the entire DNA. Here you just use a chunk for a particular protein, because let's say this is DNA, right? These genes code for, let's say, skin cells, right? I'm exaggerating right now. So this particular genes will code for hormone, for example, right? So, but the entire DNA will code for the entire human body, right? So here we're just using a little chunk to make a particular protein. So we'll make a copy of this original DNA. Here's the copy, code, and we'll take this code out of the nucleus into the cytoplasm, where we can make or produce or synthesize proteins. So here's a simplified version on both sides. So it's called transcription, translation, but we need also initiation. We need that particular signal, that stimulus. Why do we need to make a new protein? Again, skin damage, new cells. So we take the DNA original, but this step happens within the nucleus. Let's say that the nucleus is the safe, where you have the genetic information. So if I want to make a copy, I cannot give you original, so you can go outside of that office to the Xerox machine, make a copy, and then return me back the original. No, it's not how it works. So within the nucleus, which is the safe, we have this DNA, and within the nucleus we're able to make a copy. So this step is called transcription. We take an original and we'll make a copy. And that copy is called mRNA, messenger RNA. So keep in mind, T matches with A in DNA, but in the RNA, instead of T, we'll use uracil, which will match with A, right? So transcription, it's taking the original small sequence, right? Because we're not replicating the entire DNA. We're just making a dedicated protein for a skin cell, for example. And through transcription, we're making a messenger RNA. Messenger. That's a message to synthesize proteins. Then we'll take this messenger RNA through the nuclear pores. From the nucleus to this nuclear envelope right here, we will send this mRNA messenger RNA into cytoplasm. And in the cytoplasm, we're able to translate that code. And the translation will code for a particular protein. Again, it could be code for making a skin cells, or it could be code for making a hormone, anything your system desires to synthesize. So transcription, an ingredient list to synthesize proteins, and then this ingredient list would be read by another type of machinery. It's a translation. So we will translate that ingredient list into something meaningful, which is a protein. So there's another view, more precise, right, of the same process. So here's this original DNA. Then we'll take this side and we'll make a copy. I know this side, not this side, just because you see there's a 5 prime, so it should be the opposite, the 3 prime. So in DNA, A matches with T, but now we're making an RNA, messenger RNA. So A will match with U, not with T, right? And then C with G, C with G, A with U, A with U, A with U, C with G, and so on. So you're making a message, and this message will be used to synthesize a particular protein. Remember in the beginning we discussed codon. So this nucleotides, 3 nucleotides, it's a codon, and will code for a particular amino acid. These 3 nucleotides will code for this amino acid, and so on. So bunch of amino acids in a different sequence, positive, negative, hydrophobic, hydrophilic, will code A for particular protein. So it's basically from the genetic information to a synthesis of protein. So this slide is very similar to a previous slide, right? This is the simplified version, right? From DNA to mRNA, the copy, and we're making this messenger RNA. So that messenger RNA leaves the nucleus, right, and ends up in the cytosol. So let's look what's going to happen with that messenger RNA. So the first step is called transcription, right? You're making an mRNA and sending out into the cytosol, and now it has to be translated. So there we have a ribosomes. So the ribosome is a subunit that will be helping to understand that code, that ingredient list. So the messenger RNA would be stuck in between these 2 subunits of the ribosomes, And then these codes would be presented to a something called transfer RNA. So you see this mRNA with these codons, right? This is codon. And then the transfer RNA will bring a particular amino acid to match this codon. So it's called anticodon or anticodon. So let's say AUG code for methionine, GGA code for glycine, UGU code for cytosine. So it has to match, right? Anticodon A matches with U, U matches with A, G matches with C, so nothing changed, right? RNA and RNA. So, but this is the messenger RNA from transcription. And this is a transfer RNA. So it's basically transports specific amino acids to this ribosome protein for a synthesis. And in this fashion, right, so we're able to create a sequence of amino acid that will code again for a particular protein. So here's this messenger RNA and here's this transfer RNA. So within the cytoplasm, we have the available amino acids. Where do we get them from? Well, there's 20 amino acids, right? 20 available amino acids, letters in alphabet. From our nutrition, right? So the foods that we consume, we bring amino acids. We are able to synthesize certain amino acids. So 20 amino acids, 9 amino acids we must consume, and 11 amino acids your body can synthesize. So here's the available amino acids. Each shape represents a particular letter in the alphabet, amino acid. So this tRNA grabs its particular amino acid that has been designed for and brings it here, and it will match the code, right? It's called anticodon. It will match the codons on the mRNA, and you're synthesizing a new protein. So you put this letter in a different sequence and you will code for a particular amino acid, and eventually a bunch of amino acids will code for a particular protein. So here we're looking at very simplified view of how the transcription and translation happens. Obviously it's very complicated, so there's a proofread as well. There's a bunch of enzymes that participate in this process. We're not discussing that. We just want to look at the general view of how it happens. So remember, from the cell lecture we discussed ribosomes, right? So the roughy R, smoothie R. So this is the cistern on the roughy R, and so that's where the synthesis of the new proteins may happen. So remember, with the roughy R, this little dots represents a ribosomes. They're sitting right on the cisterns of the roughy R. There's also free ribosomes in cytoplasm. So basically, the free ribosomes synthesize protein that stay and function in the cytoplasm. And the membrane-bound ribosomes, let's say here, right, synthesize protein that will become components of the cell membranes or will be exported from the cell, let's say hormones and enzymes. So the cisterns is like a collecting pot that the newly synthesized protein will end up here, and then they will be packaged into a vesicles and will go wherever they need to go. So now let's recap. So here you see the DNA, right? Just one template. This is in the nucleus, and this process is called transcription. So from the DNA we'll get to a mRNA, right? T with A with U, cannot be T here because it's a RNA, right? And C with G, and so on. So we're creating a codon. This codon will code for a amino acid. So then mRNA leaves the nucleus, ends up in the cytoplasm, and then this mRNA would be squeezed in between two subunits of the ribosome, and the transfer RNA will bring a corresponding amino acid to match this. Anticodon matches U with A with U, G with C, and so on. And then you will have a particular sequence of amino acid that will code for a particular protein. I'm like a broken machine, repeating, and repeating. At the beginning, this process may look complex, but it's pretty straightforward and very simple. right? So there are 4 types of nucleotides, right? So there are 4 possible combinations. It's 4 in the power of 3, right? Because the codons is just 3 letters, right? And then there's another 3 letters. So that will get us to 64 possible combinations. And there are 20 available amino acids, and you can put them in different sequence. So that's why we can code for thousands of protein, letters in the alphabet. So basically in DNA replication, you unravel, you open up the DNA, right? T, A, G, C, so A, T, C, G, and then you make a new copy, right? You're matching with A, T, C, G, and here's T, A, G, C. You make identical copies. So it's a DNA replication. So you can make 2 daughter cells. In protein synthesis, you take a DNA, I'm gonna go like that. You take a DNA, T, A, G, C, T, A, and you're making a messenger RNA that serves as a template to synthesize protein. So T with A with U, G with C, C with G, T with A, and A with U, right? So you have your messenger, leaves the nucleus because this is transcription. It happens inside of the nucleus. You take this into a cytoplasm or on the RAFIR, right? And here's a ribosome, and then you need to translate. So here's transcription, and now it's translation. Translation. So now you have a transfer RNA will bring you a corresponding amino acid. This amino acid looks like this, and this amino acid looks like that. So this is a transfer RNA, and that will code for a particular protein, and that protein could be a hormone, enzyme, or structural part of your cell membrane. So when you look at this, it's pretty straightforward. So please know exactly what is messenger RNA, what is transfer RNA, what is replication, making identical copy because we need to make new cells, old cells die out, and this is a transcription translation, right? Dogma of life. So but there should be initiation as well. That's a stress. So know where the transcription happens in the nucleus, know where the translation happens in the cytoplasm, know what the ribosome functionality is, and know why this process is important. So now let's talk about cell cycle. So the cell cycle involves DNA replication and making 2 new daughter cells. So the cell cycle is composed of phases. G1 phase, cell growth, S phase, cell synthesis. Then there's the G2, and very important one, all of them are important, are mitosis. So the G1 phase, it's when the cell is preparing itself for synthesis, right? To synthesize, we need to double in size, double in organelles, because we need to make 2 new identical daughter cells. So then there's also a gap, right? So we want to make sure that cell has enough of nutrients, cell is big enough to divide, the DNA is damaged or not damaged, because we prepare to make a copy, identical copy of the DNA in DNA replication that we just discussed. And if it's damaged, so it must be repaired before it would be copied. Then we go into second stage, there's a synthesis. So all of this is called interphase right here. So it's a preparation for the division. So the division would be here. It's called mitosis. And this is the interphase. So then we get to the S phase, we're replicating DNA copying, and then the G phase, and again there's also a gap, right? So we want to make sure is all DNA replicated correctly, is environment is favorable to proceed further. Then with the G2 phase, we're just continuing to grow. And then we proceed to mitosis, that also broken down in subphases. But the actual steps in mitosis we'll discuss in another video. So again, this phase from the G1 all the way to mitosis is called interphase. But between G1 and synthesis, there's also G0. Cell can live the entire cycle. So they start to grow here, but then they decide, I'm not going to divide. The cell maintains its special characteristics, but those not replicated in DNA or divide. In DNA synthesis, right, there's a replication of the entire genome, of all of those 46 chromosomes. Then, again, another gap. Did we divide correctly? Everything is fine. Then we can proceed further down to G2, and the G2 is just a continuation and the synthesizing of new proteins, continuation of growing, right, doubling, because we really need to create two new identical daughter cells. And then we proceed to M phase, or mitosis. And the mitosis includes prophase, metaphase, anaphase, telophase, and cytokinesis. I'm gonna discuss mitosis separately, but quick overview. In prophase, that's where the newly synthesized DNA will be visible. In the metaphase, this new DNA, or chromosomes, or sister chromatids would line up for preparation for the division. In anaphase, we will divide the sister chromatids, and the telophase, that's where the nuclear envelope will be built around this new DNA, and with cytokinesis when it's a complete division. But we will discuss this later on. I hope it was educational. Thank you very much. Until next time.